I presented findings of a correlative study of the CHAARTED trial, specifically focusing on gene expression biomarkers. So the CHAARTED trial was a study that was reported about 6 years ago of men with metastatic hormone-sensitive prostate cancer who were randomized to standard of treatment, which is androgen deprivation therapy, so hormonal therapy, or the addition of 6 cycles of docetaxel chemotherapy. Basically what that trial showed was a significant survival advantage with early chemotherapy, which we had traditionally used for men with hormone-resistant or castration-resistant prostate cancer. Now, since that time, many new treatments have developed in that space, most notably hormone agents like abiraterone or enzalutamide or apalutamide. And one of the clinical challenges we're facing is how do we rationally choose which therapy is best for which patient? And we think that probably the underlying biology of the disease or changes in the tumours may give us a better clue than what we have clinically. So we, in this study presented at the symposium, performed RNA profiling of tumours from the CHAARTED trial. So these were biopsies taken from the prostate before men started hormonal therapy. And we classified these tumours into what we call luminal and basal subtypes. Now, this has been done previously in localized prostate cancer, where luminal B subtype has been associated with a poorer prognosis and has generally more aggressive features. In the CHAARTED population, what we noticed was that the 3 subtypes that we had seen in localized prostate cancer, luminal A, luminal B, and basal, had a different distribution. There were very few luminal A tumours, and about half the patients were luminal B and the other half were basal. Importantly, we saw that luminal B tumours were associated with a shorter survival compared to basal subtype. However, despite that shorter survival, addition of the docetaxel chemotherapy related to a pronounced overall survival benefit. Intriguingly, the basal tumours, which had a relatively more favourable prognosis on hormone therapy alone, did not show a benefit from the addition of docetaxel. So collectively, these data point to the fact that this PAM50 classification, or the luminal basal classification, may have both prognostic and predictive effects. These data require validation in independent cohorts, and so we are partnering with investigators from other trials of metastatic hormone-sensitive prostate cancer to test these findings and hopefully validate what we see, all with a view to leading to a treatment paradigm that's has more precision to it and is guided by biomarkers and biology.